Thanks, Mickey. Is this on? I guess it's supposed to be on for a recording. So it's a great pleasure to be here. I like to start my talks in Jerusalem by saying that the first time I came here was when I drove here from London in 1965 on my way to Persepolis with 3 other Cambridge undergraduates. Of course, Jerusalem was very different in 1965, and even this part of it might have been not in Israel, and I don't quite know for certain. Anyway, I've been back many times since. It's always a pleasure to visit this beautiful city. So, as advertised, for the first 30 minutes or so, I'll be giving a rather general introduction to the field of post-translational modifications, and particularly reversible modifications, using phosphorylation as a paradigm. So, phosphorylation, like many post-translational modifications, is catalyzed by an enzyme that adds the modification, in this case phosphate, using ATP as a phosphate donor, most commonly to serine, threonine, and tyrosine, creating a phosphate ester linkage. This modification then changes the state of the protein by adding a phosphate to some defined target amino acid, often in an unstructured region between domains or at N and C termini. The phosphate can be removed by a different class of enzyme, in this case the protein phosphatases, to revert the protein back to its base state. So these are called a writer and an eraser in today's parlance. And then the state of the phosphoprotein can be changed in many ways, but commonly by being recognized by a phosphobinding protein called a reader in this case, and that then transduces a signal downstream. So over 400 different post-translational modifications are known throughout life, and more are still being discovered through the use of mass spec. Phosphorylation, at least in eukaryotes, is the most prominent post-translational modification. And in HeLa cells, Matthias Mann's group showed that there are 40,000 different phosphorylation sites. So I will argue that PTMs, post-translational modifications, are a major mechanism for increasing proteome complexity. So post-translational modifications can either be reversible or irreversible. And in the case of reversible modifications, they all follow the same paradigm. There's a writer and an eraser. So for instance, for ubiquitination, which can target proteins for degradation by the proteasome, there are a series of ubiquitin ligases and a series of ubiquitin proteases, and also there are readers. So I'll just give you some examples of some new post-translational modifications that you might not necessarily have heard of. For instance, phosphoribosyle ubiquitination, discovered by Ivan Dikic's group, which modifies serine residues. And there are many additional types of esterification of lysine, like malolination and succinylation, which have appeared in a fairly recent history. And then another concept that's worth considering as we find more modifications is that there can be competition between different modifications occurring at the same amino acid. For instance, serine and threonine can be modified by a whole series of different PTMs, and there's some evidence that phosphorylation and O-glucnacylation, this addition of a single O-glucnac residue, can compete. And this might be an important regulatory principle. In addition, although I'm sure you didn't read through, 9 out of the 20 amino acids can be phosphorylated. And in the second half of my lecture, I'll be telling you about some non-canonical phosphorylations, particularly phosphorylation of histidine. So the human proteome, at least if you count the number of genes, is around 20,000 proteins. There are some alternative splice forms. which might increase the total number of protein products to around 50 to 100,000. It's a large number, but perhaps not a large enough number to regulate the complex body plan and behavior of vertebrates, for instance. And so adding post-translational modifications to proteins greatly increases the number of protein states, perhaps to several hundred thousand or a million. And particularly when used in combination, that can increase proteome complexity. And as I've already indicated, many of these modifications can be recognized by reader domains in other proteins. The first example was the work of Tony Pawson, the late Tony Pawson, who showed that the SH2 domain, these are all small modular domains, can recognize specific phosphotyrosine residues. There are readers, as I'll show you in just a second, of phosphothreonine and phosphoserine. In fact, many PTMs have reader domains, ubiquitin sumo, methyl lysine, and astyl lysine. These are particularly important in regulating transcriptional output. Having mentioned Tony Pawson, I thought I would show you this historical slide, which Tony put together shortly after the human genome sequence was completed, showing some of the modular domains that had been identified in 2003, which recognize, some of them recognize post-translational modifications like these three here. Others recognize lipids, phospholipids, others simply short sequence motifs like SH3 domains, recognizing short proline motifs, etc. So this concept then of modular binding domains is extremely important in signal transduction, as I expect you'll be hearing throughout the course of this meeting. And here's an example of how these domains work. This is the structure of the Salk tyrosine kinase SH2 domain bound to a phosphotyrosine containing peptide. So that in red is the structure of the globular SH2 domain, and you can see there is a deep pocket into which the phosphate of the phosphotyrosine is inserted, interacting with this arginine at the base, and then residues on the C-terminal side of the phosphotyrosine are also dipping into pockets on the surface. So this is a sequence-dependent phosphotyrosine binding domain. I don't expect you to read this, but I'll just point out that there are a very large number of different phosphoserine threonine binding domains. Perhaps the best known might be the 1433 family, which are involved in many signaling pathways. And there are also several other phosphotyrosine binding domains, such as the PTB domain. And this gives you a sense of the diversity of the structures of some of the phosphoserine threonine binding domains. You can see there is really no common structural motif. Many different types of structure can recognize phosphoserine and phosphothreonine in a sequence-dependent manner. PIN1 is a protein that we had worked on and recognizes phosphoserine, threonine, and proline, and is involved in isomerization of this proline bond. So phosphorylation is the most ancient of post-translational modifications. It has a long history. I normally date the start in 1906, work of Phoebus Levine at the Rockefeller Institute, who showed that phosphate is covalently linked to vitellin. It's an egg yolk protein. But some people date the discovery even earlier to 1883, when Hammerstein's lab showed that there was phosphate in casein, which is perhaps the best known phosphoprotein. It wasn't until 1932 when the nature of the phosphate linkage was identified by Fritz Lippmann, who was working with Phoebus Levine. Fritz Lippmann obviously went on to do other very important things. And at that same time, he synthesized not only phosphoserine and phosphothreonine, but also phosphotyrosine, because he had deduced that this must be linked via a hydroxyester, but didn't know which of the three hydroxy amino acids it was linked to. So he synthesized phosphotyrosine. Enzymes that add phosphate to proteins were not really discovered until many years later, another 30 years later, the work of Eugene Kennedy, who reported a protein kinase activity associated with mitochondrial fraction that could phosphorylate casein. He didn't continue to work on protein kinases. He actually worked on lipid synthesis and structure. The first enzyme purified protein kinases were the work of Krebs and Fischer, phosphorylase kinase in 1959, the enzyme that regulates glycogen phosphorylase and the degradation of or release of glucose from glycogen. And the work of Ed Krebs, the first regulated protein kinase, the cyclic AMP-dependent protein kinase in 1968. Tyrosine kinase activity wasn't reported until 1979. There's been a huge amount of progress in understanding what tyrosine phosphorylation does in terms of signal transduction since then. And one outcome of that discovery is approval of a series of drugs, as you heard from Mickey, that target tyrosine kinases, particularly for cancer therapy. So I thought it might be interesting for any students in the audience for me to tell you a little bit about how tyrosine phosphorylation was discovered. This was the autoradiogram that led me on this phosphorylation odyssey. I'm sure you all can't see the spot that intrigued me, but it's there with the eye of faith. So how did I come across tyrosine phosphorylation? Well, back in 1979, so almost 40 years ago now, I looked a bit like this. Beard was longer and darker. And I was working on polyomavirus. It's a small DNA tumor virus. It's a circular DNA genome, around 5,500 base pairs. It's split into two halves, if you like, functionally. There's an early region which is expressed immediately the virus infects a cell. And then the late region is expressed after DNA replication and encodes the coat proteins that package the virus to this icosahedral structure. The early region is the key for this virus to transform cells into tumor cells, and it expresses three overlapping proteins, the so-called T antigens or tumor antigens that can be detected with anti-tumor serum. They are the result of alternative splicing, and interestingly, actually, this region of the genome here is read in all three reading frames. Extreme genetic compaction. And we and others had shown that of these three T antigens, the middle T antigen was most important for transforming fibroblasts in culture. And so we were focused on trying to understand how this protein could transform cells, a single protein able to switch a cell from a normal state into a transformed state. So we had no real clue how this protein did this. Of course, this was really before the days when we had a proteome to compare it to. And even if we had a proteome, middle T is unrelated to any cellular protein. It wouldn't have given us any clue. But in 1978, so the year before, Mark Collette and Ray Erickson had reported that the VSark protein, the transforming protein of a different tumor virus, sarcoma virus, an RNA tumor virus, had an associated protein kinase activity. Now this was exciting because we knew that protein phosphorylation could regulate protein function and that protein kinases often had many targets, and so we could perhaps understand how this protein kinase could transform a cell by switching on and off many proteins. And so we wondered, as did others, whether this might be a common mechanism of tumor virus transformation. So we set out to test whether middle T has an associated protein kinase activity. And so the way we did this was to isolate the tumor antigens by immunoprecipitation with some anti-tumor serum we had available. And then we incubated the immunoprecipitate with gamma P32 labeled ATP to allow the kinase to work, and then ran out the samples on an SDS gel. And you can see in lane C here that only one band became labeled in this in vitro kinase reaction, and that was the middle T band, as you can see from the S35 label in lane A here. So that was obviously pretty exciting. And we actually had some mutant viruses that could not transform cells that had been derived by various groups. And you can see some of these viruses here, like NG18 and NG59, even though they have a middle T, lacked this kinase activity. So there was a correlation between transformation and whatever this kinase was. So other groups working on polyomavirus had also tested whether polyoma middle T might have kinase activity, and particularly Alan Smith and Mike Fried at ICRF in London, and Brian Schaffhausen and Tom Benjamin at Harvard. And we all got together at the 1979 Cold Spring Harbor Summer Symposium on tumor viruses in May of 1979, and found out what each other group had been doing, talked about our work at the Cold Spring Harbor Symposium, and then agreed to co-submit our papers to Cell when we got back. And our paper was submitted on June the 11th, 1979. Now, it was just as hard to get papers published in Cell then as it is now. And here are the reviews on our paper. These two findings force one to seriously consider the possibility that the activity observed in immunoprecipitates is an in vitro artifact. Not a good start. The product of this in vitro protein kinase-like reaction, phosphoserine, etc., wasn't characterized, with the very low reported activity, an adenylation reaction of some contaminant of the ATP preparation is possible. And reviewer 3 was the most damning. Unfortunately, very few of these conclusions drawn by the authors are actually clearly substantiated by the data. So you would have thought this would mean outright rejection, but in 1979, Ben Lewin, who was the founding editor of Cell, could do whatever he liked, and he asked us to submit a revised version. And all three groups got to submit us a revised. He thought this was exciting, and he wanted to publish these papers, if possible. So as reviewer 2 had said, we hadn't characterized the nature of the phosphoamino acid in middle T. And so the day after we submitted, on the 12th of June, 1979, this is the page from my lab notebook, I set out to test this by doing another in vitro kinase assay on middle T, isolating the protein, and I know you can't read this, but basically hydrolyzing it in acid, 6 normal HCl, 100 degrees for 1 hour, which releases phosphoamino acids. It basically cuts the protein into single amino acids. And that was what I ran out on that thin layer plate by electrophoresis at pH 1.9 on June the 14th, 1979. And put the thin layer plate down against X-ray film, and included in the sample were unlabeled phosphoserine and phosphothreonine, because we knew it would be one of those two, the only two reported phosphoamino acids. And you may or may not be able to see the pencil circles here. And this spot that I showed you here runs actually between the phosphoserine and phosphothreonine markers. So here we had a new spot. It was acid stable, but it wasn't phosphoserine or phosphothreonine. At the time, I laconically said the spot doesn't co-migrate with phosphoserine or phosphothreonine. So what was it? I repeated the experiment. Of course, it could have been some giant artifact, as the reviewer said, one of the reviewers said. But I got the same answer the second time I did the experiment, and as I said, whatever it was stable to acid hydrolysis. And luckily, I'd been trained as a biochemist in Cambridge, and I knew there was a third hydroxyamino acid, tyrosine. And so I guessed that this new spot might be phosphotyrosine, even though no one had ever reported a tyrosine kinase. So what I was proposing was that instead of phosphorylation occurring at the beta carbon hydroxyl group of serine and threonine, phosphorylation of the phenolic hydroxyl group in tyrosine was occurring. And so, to test this idea, I tried to make some phosphotyrosine as a marker. Now, I'm not a chemist, I'm a biochemist, and what I tried was to mix tyrosine with phosphorus oxychloride, not realizing that this would give a black tar, which it did in the bottom of the tube. But I was able to extract a little authentic phosphotyrosine from this mixture. And on July the 2nd, I ran this compound out together with phosphoserine and phosphothreonine, and it migrated between the two in roughly the spot of the radioactive compound I'd seen. So, despite the possibility I might have discovered something important, on July the 3rd, I left with some colleagues from the Institute to drive up to Idaho to raft the Salmon River. And from there I went on to Cambridge in England to attend the DNA tumor virus meeting, not getting back to La Jolla until a month later. So at the tumor virus meeting in Cambridge, I talked about the kinase activity again, but not about the possibility it might be phosphotyrosine. And here I am, rowing this boat on the Salmon River in Idaho in July of 1979. So when I got back, I had marker phosphotyrosine. We devised a better separation in 2 dimensions now, electrophoresis at pH 1.9 and chromatography, and it's quite clear that the radioactive spot from middle T co-migrates with phosphotyrosine. And we included this figure in the revised version of the paper that was sent back to Cell on September the 25th. And you'll note that it was accepted on September the 27th. So we sent it on September the 25th by FedEx. Of course, this was before electronic submission. So it didn't get there until September the 26th, and it was accepted the next day. So, of course, what this meant was that Lewin had decided to accept these papers regardless. And it turned out the other 2 groups had either not been asked to do the phosphoamino acid analysis or didn't do it. But all the papers came out back to back to back in the December issue of Cell in 1979. So you may have noticed that where I had put pH 1.9 on the slides, it was in quotes. And the reason it was in quotes was actually what had happened was that the night I did the first experiment, I was late, and I was too lazy to make up fresh pH 1.9 buffer, which contains formic acid and acetic acid, and with repeated use, the acetic acid is more volatile and is lost, and the pH of the buffer falls to pH 1.7. And it was because the pH of the buffer had fallen that I was able to resolve phosphotyrosine and phosphothreonine. Ray Erickson had reported that the target for the Salk kinase was phosphothreonine using Whatman paper and I guess fresh pH 1.9 buffer, and he had found he missed finding that Salk also phosphorylates tyrosine. And it also turned out, subsequently, Sarah Courtney showed that the polyoma middle T associated tyrosine kinase activity is not an intrinsic function, unlike the VSark protein, but is due to an associated CSark tyrosine kinase. So I had a second stroke of luck, and that was that Karen Beeman in the lab had brought the Rous sarcoma virus system and was working on Rous sarcoma virus. And so one day in doing the polyomavirus validation experiments, I used a Salk immunoprecipitate as a control because I knew it was going to be phosphothreonine, as Erickson had reported. And in this case, the Salk kinase phosphorylates the antibody and not itself. And much to my amazement, Salk turned out to be a tyrosine kinase as well. And you can see that here in this 2-dimensional separation, that the heavy chain is phosphorylated on tyrosine by Salk. And with Salk, we could do one more experiment that really, I think, nailed this conclusion. we were able to show that in racecomovirus transformed chick cells, that the virus increases the level of phosphotyrosine in cellular protein dramatically. So in this case, we developed yet another separation system in 2 dimensions, now using electrophoresis at pH 3.5 in the second dimension. So we could resolve phosphoserine, phosphothreonine, and phosphotyrosine very easily. And you can see there's about a 10-fold increase in phosphotyrosine. And this falls back to the low level in normal cells when one has a temperature-sensitive Salk protein. So we were fairly convinced by this then that Salk transforms cells by phosphorylating cellular proteins on tyrosine. So we submitted a paper describing the Salk tyrosine kinase activity to PNAS via Bob Holly, who was a National Academy member in the neighboring lab to mine. And this paper, as I indicated here, has fared a lot better. The findings are both interesting and potentially extremely significant in that phosphorylation of tyrosine is normally a rare event. The primary significance of this work derives from the fact that the phosphorylation of tyrosine is such a novel activity for a kinase. And then finally, a handwritten review. This was in the days when you had multiple copies to press on. You could write on them. No revision is necessary, and immediate publication is strongly recommended. So there you are. We published this in PNAS in March of 1980. There was no immediate online publication, and so this took, we submitted it in November, so it took nearly 4 months to come out. In the interim, 2 other groups who'd heard me speak about this had already published papers on tyrosine kinase activity in journals which published weekly rather than monthly, and so we were scooped in one sense, but not really. So amazingly, actually, all of the experiments that I've talked about in the Cell and PNAS papers were done in less than 5 months. And all of the experiments for the PNAS paper were done and repeated in less than a month. So one could actually do science pretty quickly, even back 40 years ago. And if anyone's interested, I wrote a little review, which was reviewed by Yossi as a perspective in PNAS on the timeline of all these experiments back in 2015. So by the end of 1980, we knew of 4 distinct tyrosine kinases. We knew that VSark was a tyrosine kinase. Work from Owen Whitty and David Baltimore has shown that the V-able, so these are viral transforming proteins, were both tyrosine kinases. The work of Mariano Barbacid and Chuck Shear had shown that the VFIPS-FES transforming protein was a tyrosine kinase, and Stanley Cohn had shown that the EGF receptor was a tyrosine kinase. Now, all of this was done without any DNA sequences. This was simply showing that the peptide maps of these proteins were distinct from one another, and therefore they had to be different gene products. We didn't really learn how many tyrosine kinases there were until the first kinase sequences became available. So beginning in the early 1980s, first with the sequence of the VSark gene, and then subsequently with several other viral tyrosine kinase genes, it became possible to begin to align sequences, and I was interested in doing this, and so here's a manual alignment. So BB stands for before blast here. So this was all done by eye. And it's pretty clear that the viral tyrosine kinases all had a related sequence with a number of motifs scattered along the length of the catalytic domain, which is around 30,000 Daltons. And we could show, and actually Margaret Dayhoff had originally shown that the Salk kinase and the cyclic AMP-dependent protein kinase, which interestingly, had been sequenced the old-fashioned way. In other words, the protein had been sequenced and not the DNA, were related. And so we learned fairly early on then the serine, threonine, and tyrosine kinases were all related in sequence. They had these characteristic motifs that could then be used to identify other protein kinases. And there was one sequence, this particular one, HRD, which was distinct for the tyrosine kinases in the top half of the slide and the serine, threonine kinases in the bottom half. The first official kinase sequence alignment was published in Scientific American, not exactly the journal where you would go and look for these, in an article I wrote on the proteins of oncogenes back in 1985, and then a subsequent more official alignment came out in the Annual Review of Biochemistry. The motifs that I'd identified turned out subsequently, when protein kinase structures became available, to line the catalytic cleft. So here's that RDL motif I pointed out. This aspartate here is the catalytic base. DFG is highly conserved. This chelates a magnesium ion. And here are the lysine that's in the ATP binding site that's often mutated when you want to make a dead kinase mutant. And you can see here is the ATP molecule bound in Susan Taylor's beautiful 1991 structure with the gamma phosphate here ready to be transferred to a protein substrate. So the availability of those motifs actually enable people to identify new protein kinases from the increasing number of cDNA sequences, and also to use degenerate PCR to identify new kinases. And it became clear sort of in the mid-1980s there would be a very large number of distinct protein kinase genes. So we started out with phosphorylase kinase and pKa, as I said, but you can see this sudden upturn when PCR and DNA sequencing became readily available, and by 1987 it looked like there might be, a very large number, and I predicted there might be as many as 1,001 protein kinases. And the number of tyrosine kinases was also, predicted tyrosine kinases was also rapidly increasing. So we know now that the number is not 1,001, but it's a significant number, around 540 in the human kinome, as we call the collection of protein kinase genes. But there are kinomes which are significantly larger. So the eucalyptus kinome, I'm sure there are eucalyptus trees here. They come from Australia originally. They grow in California, has 2,500 protein kinase genes, largely due to genome duplications, I would say. And paramecium, that single-cell flagellated organism that people often look at in high school, has nearly 3,000 protein kinase genes. So in general, protein kinase genes occupy about 2% of a eukaryotic kinome, all the way from yeast to vertebrates. So when the human genome was sequenced, we could survey it for protein kinase genes, particularly the work of Gerard Manning at Sugen, the Ossie Schlesinger's company. We were able to deduce how many protein kinase genes there were. And the bulk of those protein kinases are related to one another, are displayed here in this catalytic domain tree in 7 main branches with the tyrosine kinases on the top. In addition to those 478 related kinases, there are a few atypical protein kinases. Particularly important are the PI kinase related kinases here. Here they are, PI kinase related kinases here, which includes ATM and ATR, the DNA damage response kinases, mTOR, the nutrient sensor kinase, and DNA PK and other DNA damage kinase. These are very ancient kinases. They're present in yeast and in all eukaryotes. Now, at that time, we had 518 protein kinase genes. Interestingly, about 10% are missing one or more of those key catalytic residues in the motifs, and they are classified as pseudokinases. Some of them are highly conserved in evolution. They appear in many cases to serve as scaffolds. The protein kinase fold, that bilobate fold I showed you for pKa, is obviously a very stable protein fold, and so these catalytically dead kinases apparently serve to scaffold other proteins, including other protein kinases. Now, in the 16 years since human kinome 1.0, a few new kinases have been added. All of them are remote or atypical kinases that are either distant or not related in sequence to the larger family. These include some important kinases. So, for instance, FAM20C, not a very auspicious name for a protein kinase, turns out to be the real casein kinase. This is a protein kinase that lives in the lumen of the Golgi, and it phosphorylates proteins in the secretory pathway on the way out of the cell, and in particular casein, which is a milk protein that is highly phosphorylated. In addition, there is a family of protein kinases that I'll be talking about in the second half of my lecture, which are the only reported histidine kinases. So there were no additional tyrosine kinases, and I'll tell you in a second about the numbers. I should just point out before going on to tell you about the number of tyrosine kinases that any process regulated by phosphorylation requires not only a kinase but a phosphatase, as I pointed out in my first slide. And the phosphatases are a little more complicated than the kinase. There are 10 distinct structural families of protein phosphatases illustrated here. There are a total of 190 catalytic domains, so not quite as many kinase domains as there are phosphatase. Not quite as many phosphatase domains as there are kinase domains. But the complexity of the protein phosphatase family is greatly enhanced by the fact that many of them have regulatory subunits. For instance, PP2A has about 30 different regulatory subunits, and these complexes greatly expand then the diversity of the protein phosphatases. So overall, including the phosphobinding domains, probably close to 4 to 5% of the genes in a typical genome are involved in protein phosphorylation and downstream signaling. Yes, so how many tyrosine kinases are there? By the end of 1990, we knew there were over 50 tyrosine kinases based simply on cloning new tyrosine kinases. But with the completion of the human genome, we could say there were 90 tyrosine kinases. Here is that branch of the tree here. Here is Salk, the first tyrosine kinase, with its 7 family members here. Many of these branches are of receptor tyrosine kinases. So this is the FGF receptor branch that Yossi will be talking about. The largest family of receptor kinases is the EPH family here that are involved in development, particularly formation of boundaries in tissues. There are 13 of them. And the EGF receptor is over here. Over half of these kinases are implicated in cancer through activating mutations or overexpression. And if we look at the whole kinome of the 535 or so human protein kinases, possibly up to 200 are involved in disease, most of these in cancer. But we know of several other kinases that are mutated, sometimes in hereditary diseases, in other cellular organismal processes. The finding that tyrosine kinases were able to drive cancer, first with viruses and then subsequently cellular genes in cancer, led to a lot of interest in the development of drugs that target these tyrosine kinases, both at the catalytic level with small molecules or antibodies. And currently there are 45 protein kinase inhibitors approved for therapy, and 32 of these are tyrosine kinases. And here in a slide put together by Doriano Fabro, you can see the progress in the development of kinase drugs. And starting out in 2001, imatinib or Gleevec, which targets BCR-ABL, was approved, and you can see there's just been a steady increase in the number of kinase inhibitors that have been approved. And sunitinib, which I can't see down here, was generated by Yossi and Schlesinger and Axel Ulrich's company Sugen, and was one of the early ones to be approved. There are many, many more tyrosine kinase inhibitors and also serine threonine kinase inhibitors in clinical trials, and this number will continue to increase over the coming years. So let me finish then this first part of my lecture by pointing out to the students in the audience that progress in science can take a long time. So CML, the disease which is targeted by Gleevec, was discovered in the mid-1800s here. It was shown to be a unique type of leukemia, chronic myelogenous leukemia. A chromosome, a marker chromosome in 1960 by Noel and Hungerford was identified, which was missing part of chromosome 22, and then shown by Janet Rowley in 1973 to be a reciprocal translocation, which fused or brought together the BCR gene with the ABL gene that encodes the BCR-ABL protein that was shown to have high tyrosine kinase activity because BCR oligomerizes ABL. So shown to cause CML in mice. In the interim, Rous-sarcoma virus had been identified in 1911 by Peyton Rouse. The Salk gene identified in 1970 and shown to be derived from the C-Salk gene by Bishop and Varmus in 1975. Protein kinase activity, as I told you, by Colette and Erickson in 1978, and then the fact that it's a tyrosine kinase in 1979. And then there were other strands of research going on at the same time that contributed to this. But all of this was really needed before the first tyrosine kinase inhibitors were developed. Initially in academia by Alex Levitsky, who will be speaking later, and then through pharma. In this case, it was actually Siba Geigy who developed what became Gleevec, CGP57148, initially as an inhibitor of the PDGF receptor, but later shown to inhibit ABL, and this was what became STI-571, imatinib, and Gleevec. And so from the time of the discovery of CML to the approval of the first drug was nearly 150 years. So progress can take a long time, but I thought this was quite interesting history. So with that, I'll end the first half of my talk, and if there are any questions, I'm happy to take them before I go on to talk about some of our recent research. Yes, Bob?
Your story about the pH of the buffer is one I've told many times.
Good.
Very student group. As sort of a quintessential example of the role of serendipity in science. Now, but I want to clarify a couple of things to make sure I haven't exaggerated. As I tell your story, at some point, you had trouble yourself reproducing the finding in your laboratory because you were using pressure buffer.
Yeah, what I didn't say was that before we published this, I thought I should do the right thing and make up fresh pH 1.9 buffer. And when I did that, I realized that phosphotyrosine no longer resolved from phosphothreonine. And
Did that worry you at the time?
yeah, it worried me a lot. And so for the next 2 days, I ran the buffer over and over again and eventually recovered the separation, and I realized it was due to a change in the pH of the
Just
buffer.
didn't measure the pH? You should compare the pH.
It wasn't the obvious thing to do initially.
And so, isn't it true that a number of very important groups at that time using the correct buffer had misidentified?
Yeah.
Which I remember
Stanley Cohen had misidentified it, yeah,
completely and published that.
and published it as a
Using the right
phosphothreonine, right, yeah.
So Tony, when you showed 2D chromatograms or electrophotograms later, and you marked it also 1.9, it was still using 1.7.
The first 2D separations were done with old buffer. Yeah, we discovered how to, the trick was the old buffer. Yeah. It turns out that Stanley Cohen eventually showed if you use pH 1.7 buffer, which is just formic acid, you get a very nice separation. And so, it was definitely a lot of luck, serendipity. Chance favors the prepared mind, perhaps, since I knew tyrosine was a third hydroxyamino acid. Any questions from students about serendipity in science? I mean, what I would say is, when you get an unexpected result, first thing to think is probably it's an artifact. I should repeat the experiment. But if the result is reproducible, then don't necessarily discard it. Think if there might be some important reason why you've got something that goes against what has been published, for instance. Oops. Okay.